All right, so today we are looking at Chapter 8, which is going to focus on DNA replication, which is the process of taking a copy of your DNA and making an exact identical copy of it. We're also going to talk about mitosis, which is the process of cell division. We're going to talk about the importance of that. And then we're also going to talk about the cell cycle, which is kind of this internal clock in your cell that tells your cell when it's time to divide and when it's not time to divide. And so we're going to start off looking at the two different types of cell division. And there are two types of cell division for two different types of cells. The main type of cell division that your cells are going to do are called mitosis. And so mitosis is the process of starting with one cell, and then after mitosis you end up with two cells. And these cells are going to be exact genetic clones of the original cell. And mitosis is going to happen in all of your body cells, every single cell except in your gametes. So if you're male, that means your sperm, and if you're female, that means your egg. So there's not going to be mitosis occurring in the gametes. But all other cells of your body, skin cells, stomach cells, heart cells, liver cells, epithelial cells, are all going to undergo mitosis. Now, meiosis is going to be the other type of cell division, and meiosis is the division of the gametes. But meiosis is going to be a little bit different because you're going to have two cell divisions. So after the second set of cell divisions, you're going to end up with 4 cells. And so these 4 cells that you're making are going to be the gametes. If you're a guy, you're making sperm in meiosis. If you're a female, you're making an egg in meiosis. So meiosis is the process of creating these sperm or creating these eggs. And so these are only going to happen in these cells, in these gamete cells. Whereas mitosis is going to happen in every other single cell besides the gametes. And so we're going to focus on mitosis today, but the next lecture on Monday will be more on this meiosis process. But basically there's this alternation of these two processes in human development and in several different organisms actually. And so when we start off, we start as a little baby zygote, and that's a result of, mom and dad love each other very much. And so they do the dirty, and you end up fertilizing an egg. So in fertilization, you're taking this sperm and this egg, and each one of these, I can't recall if we mentioned haploid and diploid before, but there are haploid cells and there are diploid cells. In diploid cells, that means you're going to have 2 sets of genetic information. In other words, one from mom and one from dad. In a haploid organism, you're only going to have 1 set of genetic information. So another example, humans, we have 46 chromosomes. All right, we've got 23 from mom and 23 from dad. So we are diploid organisms because we have 23 chromosomes from our mom and 23 chromosomes from our dad. There are some organisms that have even more, like bananas. They have 4 sets of genetic information. Don't ask me how that goes. But in meiosis, what we're doing is we're producing these gametes, and these gametes are going to be haploid, meaning if you're a male, you're only going to have your own DNA in there. If you're a female, then you're only going to have your DNA in the egg. And so by making these haploid gametes, when they fuse together in fertilization, so if you have 1 set of genetic information in your sperm and 1 set of genetic information in your egg, and they fuse, now your zygote is going to have how many sets of genetic information? 2, right? So we go from being, or not us physically, but the sperm and the egg go from being individual haploid organisms, and then when they fuse in fertilization, we now have a diploid or a zygote that has 2 sets of genetic information, 1 from mom and 1 from dad. And so after the fertilization occurs, we start developing from this one little diploid cell. And from this point on, we're growing and developing through mitosis. Because mitosis is just the process of dividing most of the cells of your body. Again, anything except for your sperm and your eggs. So your body's going to constantly undergo mitosis. Our cells are undergoing mitosis right now, and it will be for the rest of our life. Because that's how we grow, that's how we develop, that's how we repair tissues. And so after several years of development and mitosis, we grow up to be adults. And then once we hit puberty, we start undergoing meiosis, or our germ cells, our sperm and our eggs start undergoing meiosis, I should say. And so after meiosis, then you end up developing your sperm and your egg again, and then the cycle can kind of repeat. So there's kind of this alternation of mitosis versus meiosis. But like I said, we're going to start with mitosis today and then focus on meiosis on Monday. And so before we can divide our cell, we need to make a copy of our DNA. So if I have, let's just say 4 pieces of DNA in my initial cell, what would happen if I didn't replicate my DNA, if I didn't make a copy of it? Then after that cell division, I'd only end up with 2 chromosomes in my new cells that I just made. But since this cell has 4 chromosomes, don't you think it makes sense that the next should also have 4 chromosomes? Because we're making a genetic clone, an identical copy. That's all we're doing. We're making a copy of this cell so that you end up with 2 cells. So in order for both of these cells to get the necessary DNA, you have to make a copy of that DNA. And then through the process of mitosis, you divide those copies from one another. And you divide them into opposite sides of the cell. And then eventually that one big cell turns into 2 individual cells. So you're separating, the purpose of mitosis is to separate these replicated pieces of DNA from one another. So that way, at the end of mitosis, you end up each with a full set of genetic information. And so before we can undergo mitosis, we have to undergo this DNA replication. And DNA replication is literally just making a copy of the DNA using those complementary nucleotides. So y'all remember how we made RNA from DNA? Well, making DNA from DNA is even easier. So all you do, well, let me show it on here. What's going to happen is, remember, our DNA is in a helix. It's all like twirled around one another. So what's going to happen during replication is that DNA will kind of unfold or unwind a little bit. And then there's going to be an enzyme that's going to look at each strand. So here's an individual strand, and so that enzyme is going to make a copy of DNA that's complementary to that strand. So if this enzyme sees a G right here, what's it going to put on the new piece of DNA?
C.
A C. What about here?
A
A T, right? And then another G, and so forth and so on. So all you're doing is you're starting with a double-stranded piece of DNA. That double-stranded piece of DNA is going to be separated, and then based on complementarity, you're going to generate a new strand of DNA. So you start with 2 strands of DNA, and then you end up with 4 strands. So you've essentially made a copy of this piece of DNA. It's just in the complementary form. So if we had a sequence, G, 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 A, T, C, then when we're making our DNA, of course this would be 5' and this would be 3'. Our new strand is going to start in that, that opposite orientation because DNA is anti-parallel. So when you make your new piece of DNA, what do you think it's going to start with? C, 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 right, T, A, G. 5'. Boom, you've just replicated some DNA. Right? Simple enough? All right, now what we're going to go into is we're going to talk about some of the enzymes that are responsible and exactly how this happens. But first, I want to talk about the nature of this DNA replication. It's called semi-conservative because after the first round of DNA replication, you end up with one old strand, or one of the original strands, and one of the new strands. So it's conserving the initial piece of DNA, but only somewhat, because you end up with a new copy of DNA locked on with it. So we call it semi-conservative. If it was conservative replication, that would suggest that we're keeping both of these and that your new strand would look like this. But that's not what happens. And so that's why this is termed semi-conservative, because after one round of replication, your DNA is going to have one old strand and one new strand. All right? Make sense? Okay, questions? All right. Oh, and by the way, I know we've talked about this before, but all of our DNA is arranged on these chromosomes, and it's these chromosomes that we're going to be shifting around when we undergo cell division or undergo meiosis. But all of those chromosomes, every single gene in your body is going to consist, or is going to be made, is what your genome is made of. So your genome is every single gene in your body or in your cells. In humans, we have about I think it's like around 32,000 genes at this point. So there's lots of different genes that are making lots of different proteins. And so this DNA replication process is going to be necessary before our cells can make a copy of themselves, or before they can undergo mitosis. Okay, so we mentioned that there are some enzymes involved in this DNA replication process. It seems easy enough, right? You just plug in the complementary nucleotide. Well, there's lots of different enzymes that have to prepare the DNA to be replicated. And there are 6 of them that you need to know. And so the first thing that's going to happen, remember our DNA, it starts as a double helix, right? Well, in order for all of these enzymes to get in and do their thing to replicate your DNA, you have to make some space. So this is where we unwind the double helix. So we started off with the double-stranded, and then we separated them. So the enzyme that's separating the 2 strands from one another is called helicase. Any term that ends in ASE is going to be an enzyme. So you can see all of these, X-1, because it's not an enzyme, but all these DNA polymerase, ligase, helicase, primase, they're all enzymes. And so if you look at the title, it could give you kind of a hint on what the job is doing, or what this enzyme is doing. So the helicase is there to unwind the helix. So that's the first thing that's going to happen in DNA replication. The next thing that's going to happen is, because we have these now separate individual strands of DNA, they're going to want to go back to their natural state, to their helical state. So you have to use some proteins to help stabilize those individual strands so they just don't go back and just wind back together into a helix. And so those proteins are called binding proteins. More specifically, they're called single-stranded binding proteins, because they're stabilizing each of the single strands of DNA. And so you're going to have, the helicase is going to come in, it's going to unwind the helix, and then once you have those single strands there, then these binding proteins are going to come in, and they're going to make sure that it doesn't go back to the helical form. After that, you're going to have an enzyme called primase, which is going to make a primer. Now, what a primer is going to be a nucleotide sequence. It's usually only going to be like 20 to 30 base pairs. So when I'm talking about base pair, I'm talking like a G bound to a C, or an A bound to a T. That's one base pair. So imagine 30 of those complementary nucleotides being in a primer. And so the purpose of the primer, and by the way, this primer that we're going to be generating is going to be made out of RNA. And so the enzyme primase is adding in an RNA primer. Now that point of that primer is a landing pad for the enzyme that's going to be doing all the replicating. And so the enzyme that's doing all the replicating is called DNA polymerase III. But before DNA can start replicating your DNA, it has to have a place to bind to the DNA. And that place is going to be this RNA primer. And so without this RNA primer, DNA polymerase doesn't have any spot to join to the DNA.
Kind of acts as an active site?
I'm sorry?
Kind of acts as an active site then for...
Active site, you know, it's a risky term because you're talking about enzymes and stuff, but it's kind of like, think about, and I don't want this to confuse you, but think about when we talked about transcription. We had the promoter, which is where the RNA polymerase landed and got ready to go. This is kind of like that. It's not part of the actual DNA, but it's just a place for DNA polymerase III to land and get ready to start replicating. Daniel, question?
I'm trying to think if that answered my question. I pretty much want to know what the primer is.
Okay, so let me just, let's make some room, and I'll give you an example.
Is it just like it connects?
It's just a sequence of DNA, that's all it is. It's just a sequence of DNA, or actually, I lied, sequence of RNA. But that's all it is. So we have our strand of DNA right here, and we want to replicate it, right? So helicase has already come in, unwound the helix. You got your binding proteins on here stabilizing the single strand of DNA. And then this primase enzyme, it's going to come in. So here's my single strand of DNA. So if my DNA, let's just say it starts with ATGCCA. So what primase is going to do is primase is going to come in here, and at the beginning of the piece of DNA that you're beginning to replicate, it's going to start making a primer. And this primer, it's just a nucleotide sequence, and it's going to be complementary to the DNA. But since it's RNA, we're not going to have any T's in it, right? And so this primer is going to be complementary to the DNA. And so if your DNA, goes out, let's just say it goes out 100 base pairs, then your primer is going to stop at about 30. So it's just kind of a get you going type of thing to help the enzyme know where to start replicating the DNA. And so this primase enzyme, it's going to make a primer, an RNA primer, that's complementary to the DNA sequence. So our RNA primer would be UACGGU. And it would go on because it's a little longer than that, but for the sake of simplification, let's just say that's our primer. Now that primase has put our RNA primer in there, now the next step is now this DNA polymerase, which is the guy that's doing all the work in DNA replication, now it has a place to land because it can't bind single-stranded DNA. It can only bind double-stranded DNA. But we're using an RNA molecule. I know that's a little confusing. I'm trying not to confuse you. Just know that in order for DNA polymerase, the enzyme doing all the work in replication, it has to be able to bind to one of these RNA primers. If there's not an RNA primer, then DNA polymerase isn't going to bind, and you're not going to be able to make copies of your DNA. Yeah, Alexandra?
I'm trying to understand it, but I'm having a hard time. Is that kind of like whenever you're doing the amino acids and it has to have like something to make it bind to get the amino acid?
No, the best example that I can talk about is what happens in transcription. We have a gene which is made up of DNA, right? And so here's what's going to be, here's our gene made up of introns and exons, whatnot. Upstream, there's that promoter region. It's not part of the actual DNA. I mean, it is the DNA, but it's not part of what's going to eventually become protein. It just serves as a beginning or a start site for that RNA polymerase, which is used to make RNA in transcription. So that's as close of an analogy as I can get. So just think of this primer as like a start site, like a landing pad. It's like, getting ready to start replicating the DNA, but it has to go somewhere and get there and get ready before it can actually start replicating the DNA. And so where it's getting ready, is at this primer site. So you have to have a primer so that DNA polymerase 3 knows where to bind the DNA. Like I said, that RNA tells it, hey, come over here. We're going to start replication right here. And the reason why it's RNA instead of DNA is because the DNA polymerase can actually recognize the RNA as being different from the DNA. So that's what tells him, oh, here's my piece of RNA. I'm going to come over here, I'm going to sit on the primer, and then I'm going to get ready, and then once I get the go-ahead, then I'm going to start making copies of my DNA. So again, the reason why it's RNA is so that the DNA polymerase can recognize the RNA and say, oh, okay, that's where I'm supposed to go. If it was a piece of DNA, it's just going to look like all the rest of the DNA, right? So it needs to be different from the rest of the DNA, which is why it's an RNA primer. And so again, just kind of think of it as, here we have, let's just say that's the start line. This is behind the start line, kind of like what we talked about in transcription. Everything's getting ready to go before you actually proceed through the process. And so kind of this preparation for DNA replication is going to involve helicase, which is unwinding the helix, these binding proteins that are going to stabilize those single strands of DNA, and then primase is going to come in and make that RNA primer. And then once DNA polymerase 3 comes in, it can start going ahead and filling in the complementary nucleotides and making a copy of your DNA. And so DNA polymerase 3, again, this is the guy that's doing most of the work right here. After DNA polymerase 3 has gone down the length of the DNA and has made a copy of it, now we have this piece. So here's our original piece of DNA, and here's our new piece of DNA.